Welcome to this session. It is a pleasure for me today to introduce the keynote, Monica Chiogna. Monica is full professor of statistics at the University of Bologna, but she stayed a long time here in Padua, where she started her career obtaining her PhD in 1993. After a couple of years in Glasgow as a postdoc, she came back here in Padua as a researcher, and then in 2015, she became full professor. She has been the director of the PhD School of Statistics for many years, mentoring many talented and brilliant students, including our local organizer, Davide Rissa. As a statistician, her research interests cover a broad range of different fields, starting from computational statistics, environmental statistics, and to biostatistics. And specifically, she contributed to the development of statistical model for the evaluation of tests and biomarker for disease screening, prognosis, and risk prediction, specifically in multivariate setting. And in the last 10 years, she focused on the use of graphical model as a probabilistic tool to model biological data and to study their perturbation. And this is the topic of the keynote of today. Monica is going to tell us how graphical model can provide an efficient methodological ground to link biology and statistics. So thank you very much, Monica, for being here. And when you are ready, the stage is yours.
Thank you very much. Thanks, Chiara, for the introduction. Thanks to the organizers for this invitation. I was very pleased when I was asked to give a talk to this huge conference in the end. Also because this is a rather new community to me, so this is probably the reason why I decided to focus my talk on connections, which in my mind at macro level are connections among disciplines and in particular between statistics and biology. There is a mutual interest for the two disciplines to get in touch because biology nowadays has become a data-intensive science, pushing for the development of data analysis tools capable to improve understanding of complex biological processes, which are nowadays measured at a high dimensional level. On the other side, statistics, beside being a standalone discipline, also develops in response to scientific needs. So there is a sort of convergence of interests in the two disciplines to get together. But for this exchange to be profitable, a common language is essential because mutual understanding acts at different planes, at different levels in different disciplines. For example, a biologist understands gene transcription perfectly by identifying transcription factors, binding sites, all these genes that get activated. Now, all these biological details do not mean much to a statistician like me because we focus on random variables, probability distributions, their characteristics, quantification of sources driving the systems. So, a common ground for an exchange of conceptual understanding is needed. And in my experience, this ground exists, and it is that of graphs, which are commonly used in biology to represent biological knowledge in the form of pathways, for example, in the left-hand side, but they are also used within the statistical community in the framework of graphical modeling to represent multidimensional problems and features of their joint distribution, which is that blanket that covers all variables describing their functioning. So with some friends some years ago, when we spotted this similarity, we said, okay, why don't we try to exploit this similarity? And as pathways are the best representation of biological knowledge, which is also experimentally validated, why don't we try to import this knowledge into the statistical tools within the framework of graphical models. Now, there are various statistical techniques and tools that make use of pathways, but they do not fully exploit their topology, or maybe they only partially exploit their topology. What we wanted to do was pushing this transfer to the largest possible extent. So what I'll be telling you today is this. I'll be giving you some essentials on graphical models, really basic things to the benefit of people who are not very familiar with this modeling framework. And then I'll be showing you how we connect biological networks to graphical models and how we use graphical models to answer biological questions. And then I'll be showing you some enhancements. Now, one of the key concepts in statistics in multidimensional settings is that of conditional independence. I'll be using the smallest example possible, 3 variables, x1, x2, and x3. We say that x1 and x3 are conditionally independent given x2 if the law of x1 given x2 and x3 is equal to the law of x1 given x2. Now, the law is a pretty old-fashioned name, I suppose, for probability distributions, but it is rather more inclusive, so I prefer it. And yes, and also, moreover, I'm also an old-fashioned statistician, so it fits with me. But intuitively, conditional independence means that knowing x2 renders x3 irrelevant for explaining or predicting x1. So you can immediately have an intuition of the importance of this concept in multidimensional problems. It allows to focus on what matters and leave out what it doesn't. And by the way, this is the reason why conditional independence is so much used in artificial intelligence, because it mimics the way in which our brain functions pretty closely. We as humans are very capable to focus on what matters and rule out what it doesn't. Formally, the conditional independence allows to factorize the joint distribution of our variables into factors defined on smaller sets of variables. And here, once again, you see its usefulness. It breaks down the problem into smaller problems. There are multiple factorizations available which somehow reflect the symmetry of the concept of independence. So in our running example, for example, you can factorize the joint distribution into a factor which is essentially the joint distribution of the first 2 variables, given the conditional distribution of the third, given the second, or you can reverse the viewpoint and factorize using the joint of the last 2 variables, given the conditional of the first, given the second. Now, multiplicity of these factorizations will become relevant in the rest of this talk. One nice thing is that conditional independence can be represented graphically with graphs where each node is associated to a random variable and nodes are joined by edges which can be undirected, directed, or even bidirected, although this is a case not covered in this talk. So, for example, this is a toy undirected graph over 3 variables having 3 nodes, 1, 2, 3, connected by edges, 1, 2, 2, 3. And although it is a toy example, it gives me the possibility of introducing some technical jargon, because graphs can be broken down into graphical grains, which are cliques and separators. So cliques are subsets of nodes all joined by an edge. So we have 2 cliques here, 1, 2, and 2, 3. And separators are nodes whose removal breaks down the graph into subgraphs of smaller dimension. Here, the removal of 2 breaks down the graph into 2 subgraphs of 1 node each. Now, how do we read conditional independence of these graphs? Now, conditional independence is essentially related to the idea of blocking. Here, if we consider our running conditional independence, we can see that 2 blocks all paths between 1 and 3. So there is no way of traveling from 1 to 3 without passing through 2. So 2 is our conditional variable that separates the remaining 2. And the factorization that we saw before, following this conditional independence, can be also read off in terms of graphical grains. So factors defined on the joint distribution of clicks and on the conditional distribution of clicks given the separators. Now, of course, this is a very, very small graph, but in larger graphs, this idea of blocking gives rise to various conditional independencies that go under the name of global, local, and pairwise. So at a global level, two subsets of variables which are graphically separated by a third set are conditionally independent given the separating set. Which implies at a local level that a node like 5 is conditionally independent on all the remaining nodes like 1 and 3 given the neighbors, which are 2 and 4, which at a micro level implies that two disconnected nodes like 1 and 5 are conditionally independent one another given all the remaining nodes in the network. Now, the converse implication is not always true, but this, it is not relevant to the aim of this talk. Conditional independencies can be also represented by directed graphs, provided that there are no cycles, okay, so no arrows, no loops in the network. Here, any node is conditionally independent of its non-descendants given the parent. So, for example, here, 3 is independent of 1 conditionally on 2, which is its parent. And the factorization of the joint distribution still holds and involves the families defined with node and its parent. So it's the product of the conditional distributions over each node given the parents. Now, it is probably worth pausing a while on the meaning of arrows, because here is where biological and statistical understanding could differ. An arrow embodies the idea of a non-symmetrical relationship between nodes. There is one node coming first and another one following. Now, statistically speaking, this is a pure artifact. Although arrows play, of course, a role in the formal syntax of the model, they do not find any direct counterpart in the real world. So they only contribute partially to the interpretation of the model. And indeed, there are many DAGs that represent the same set of conditional independencies. For example, here, these 3 DAGs, they all represent our running conditional independence, and they are equivalently good to this purpose. And indeed, they are called equivalent DAGs. Now, a totally different perspective is that of causal DAGs. Here, if you imagine that a node causes the descendants, DAGs are no longer exchangeable. So, for example, here, if you imagine that in some sense 2 is a common cause of 1 and 3, the 3 DAGs we saw before cannot be exchanged, so they are no longer equivalent. Of course, you need some external information to say that 2 is a cause of 1 and 3, and you can immediately foresee the difficulties in spotting these situations only with statistical analysis. So, end of this crash course in graphical models, very quick, hope not too annoying. Let me show you how we link now biology and statistics. The first step that we take is that of converting a biological network into a graphical model. And the closest form of graphical models that we have is that of DAGs, directed graphs. So gene products in the biological pathways become variables in the graphical model, and interactions become edges. Now, if you have any familiarity with pathways, I'm sure you do better than I, you immediately spot that this conversion is not trivial at all, as biological annotation posits all sorts of complications, as discussed by Georgilovich and colleagues. Different type of relations, loops, compounds, catalysis, etc. But the good news is that it is always possible to convert a pathway into a DAG. And there is an R package, Graphite by Sales and colleagues, that does this task perfectly. So once you have a DAG, the nice thing is that answers to most biological questions can be seen as the outcomes of two actions that can be taken on a graphical model, which are the actions of doing and saying. Now, doing and saying are terms that appear in some fundamental papers of the statistical literature. They weren't created by us. And doing involves essentially applying a disturbance to a system to study its effect, typically an external intervention. Saying involves a kind of passive observation of the system in its natural state, so kind of a steady state. And typically this observation comes in different experimental conditions, and the interest is in comparing the conditions. So I'll be now giving an example of doing and saying to show you how statistical models can be used to answer biological questions. So let us start with doing. Gene silencing is a doing action, for example. In gene silencing, the expression of a gene is switched off or knocked down to study the effect typically on a phenotypic scale, okay? And it is an extremely powerful experimental technique, and it is probably just the case to recall that the first RNA interfering based therapy was approved by US regulators in August 2018. But despite this potential, experiments are very difficult to perform. For technical reasons, so the ones we all know, like for example, broadly speaking, measurement error. But these are not the only reasons. RNA molecules, which are designed to match and destroy a given gene, can also act off target. So experiments might not be as specific as desired. And what we can do here is we can sort of synthetically silencing one gene to explore the potential effects of silencing. So without performing the experiment in vivo. This might be useful to explore the effects and maybe design more specific experiments. The pipeline foresees two steps. One is the retrieval of a DAG and the estimation of the corresponding statistical model. And the other one is performing statistical silencing, which in the statistical literature is known as intervention analysis. I'll try to make myself clearer with a little example on Drosophila melanogaster. Here we have 12 genes measured, all belonging to the WNT pathway. And the interest is in silencing one of them, the naked catechol gene. Now, the genes are colored red in the biological pathway on the left-hand side, and on the right-hand side, you see the DAG, which was extracted by pathway conversion through graphite. So somehow you have to imagine to marginalize the pathway over the variables which have been observed, because of course statistics works at an observational level, so we need data, we need measurements. We can't really speculate at a theoretical level. But one interesting thing is that DAGs that are extracted by pathways through these sort of manipulations do not fit the data very well, typically. And there are various reasons for this. One is, of course, technical artifacts and, measurement error, broadly speaking. But there are more subtle reasons that I've tried to illustrate in this slide. So assume that, the true DAG is one on the left-hand side, so the little DAG with 3 nodes. And imagine not to observe 2. So you have measurements only on 1 and 3. Now, at an observational level, if you measure the association between 1 and 3, what you will find is that 1 and 3 are statistically associated because they depend on the same common factor, although this is not observed and it stays latent. So DAGs which are extracted by pathway conversion fail to annotate statistically significant associations among variables, but we need them to estimate, potential effects of silencing. So one idea is, of course, to estimate the DAG from scratch, so ignoring the a priori information given by the pathway. So do the so-called structural learning, as it is called in the machine learning community, which is a very challenging task, especially with high-dimensional graphs. Georgilovich and colleagues showed that these DAG reconstruction greatly benefits from the knowledge of a topological ordering of the variables. Now, a topological ordering is simply a sorting of the variables. So you simply have to imagine to align nodes on a line and then join them with the arrows which are annotated in the DAG. You have a topological ordering if you do not have any arrow going backwards. There are various topological sortings that can be associated to the same DAG, but any of these would do to our aim. So the idea is to throw away the DAG, retain the topological ordering, and estimate the DAG from the data using one of the approaches that the machine learning community made available. We have found that K2 type algorithms are very powerful to this task. K2 type algorithms simply incrementally add parents to a node by respecting the topological ordering, maximizing a statistical criterion. So, for example, parents of 4 are searched among 1, 7, and 2 by maximizing a proper criterion. And on doing that, this is the result we obtained in the Drosophila melanogaster case. So on the left-hand side, you have the pathway DAG, and on the right-hand side, you have the refined pathway. And you see the two structures differ quite a lot. And but even inconsistencies in these representations are interesting by themselves because they might act as pointers in which the biological annotation is not totally precise. For example, here we discovered, as you can see, that DALI and PONT are associated. One is the entry of the pathway DAG and one is the exit. And indeed, we found out that there is a feedback loop which is not annotated in the biological pathway because DALI is itself regulated by the WNT pathway. The loop is spotted by the statistical analysis and taken into account in a form which is compatible with an acyclic perspective, which does not allow loops. So there is simply an arrow going from DALI to PONT. Now, the arrows you see in the refined pathways represent statistically significant associations. So they do not have a causal interpretation like they might have in the pathway DAG. But these are the associations that we need to make a prediction at an observational level. And now we are ready to silence NKD and to evaluate the effects of this silencing, which are evaluated by measuring the so-called silencing effect, which is the difference on the mean of the variables having the value of the intervened variable set to alpha plus 1 instead of alpha. So it's a comparison of means for given values of the intervened variable. Of course, it is an estimate, so you should take into account that this quantity is prone to sampling variability. But here, all the statistical theory comes in to help, because there are various ways of taking into account for the uncertainty due to sampling variability. For example, we used Bootstrap to account for uncertainty related to the estimation of both the graphical structure and the silencing effect. And you might also resort on some sort of regularization if the sample size is so low that you cannot estimate the parameters of the statistical model properly. Now, one thing that we very much like to do is that of trying to biologically validate the tools, the statistical tools that we develop, to see whether they truly respond to the biological questions they were developed for. So here we ended up silencing NKD, physically silencing NKD. So what we have are 14 knockdown and 14 control samples and measurements of the 12 variables. And what we wanted to see was simply if predictions obtained by synthetic silencing were coherent with what we observed after the physical silencing in terms of means. And this is what we observed. Now here, blue nodes represent successes. So, you know, the predictions were coherent with what observed, and red nodes represent failures. And you see in the refined network, we are doing pretty well. We fail on Daly and Pond, which are the two variables involved in the loop, which of course cannot be dealt with properly within this framework. The same does not happen if we use the pathway DAG as a skeleton. So if we play the same game using the pathway DAG, because of course, as I said already, this does not represent observational associations very well. But in the end, we were satisfied about, you know, the pipeline for synthetic simulation. So this was an example of doing. How about saying? Now, differential expression analysis is a seeing action. You know, you can imagine diseases to be modeled as network perturbation, so shocks that affect some targets, which then propagate the shock in the network through the network connections. So typically what you have here are observations in a controlled situation, healthy individuals, say, and then you have observation in diseased individuals. And what you want to do is to compare the two conditions, so to try to explain the differences. Now here what happens is that if you marginally compare the individual variables, the gene expression, or even if you marginally compare subsets of variables, you will find them to be dysregulated. And this is correct because they probably indeed are dysregulated, but these dysregulations are probably secondary dysregulations following the initial shock that caused a disturbance to the initial targeted genes. What we really would like to do is we really would like to get as close as possible to the variables which were initially shocked. So to rephrase in statistical jargon, what we really would like to do is to get as close as possible to those variables whose differences in the marginal distributions cannot be explained by other variables in the network. So, you know, explanation means conditional independence, which means graphical models. So here, graphical models can really help, provided that a definition, a formal definition of the variables which were initially shocked is given. So we call this set of variables the source set. And the definition was formally given by Salviato and colleagues in a paper published last year. Intuitively, the source set is the smallest set of variables explaining the difference in the two conditions. So the smallest set of variables whose marginal distribution differs in the two conditions, but conditionally on whom all the laws of all the remaining variables are identical in the two conditions. what we can do here is we can provide an estimator and an estimate, of course, of a superset, the smallest superset, which is the set covering the source set, which is compatible with the graphical grains, clicks, and separators of the graphical model associated to the system. I'll be giving you just an intuition here, but if you are interested in this topic, please do follow Elisa Salviato talk later on, because she will be giving the details with greater emphasis. The pipeline is similar as before: 2 steps, retrieval of a DAG and conversion to an undirected DAG. This is a new step. And then decomposition of the statistical model and estimation of the source set. Now, here it is more convenient to move to an undirected perspective, because the problem is formulated in an undirected perspective. We have 2 conditions. We want to treat all variables on equal footing. We do not want to be influenced by loops, feedbacks, and so on. And technically speaking, these are rather more technical detail, but moving to an undirected graph somehow enlarges the statistical model. So the statistical model is able, let us say, intuitively to deal with more peculiarities than the DAG. And it is always possible to convert a DAG into a graphical model, an undirected version of it, consistently through 2 steps, graphical manipulations, which are called moralization and triangulation, which of course have a very precise probabilistic meaning, but I'm not stopping on this right now. If someone is interested, I can tell you something later maybe. What it is important is that you end up with an undirected model, which can be broken down into clicks and separators, and whose joint distribution can be factorized accordingly. So in our toy model here, for example, we have 5 clicks, which are represented in the layers, which are joined together by the separators, which act as interlocks between subsequent clicks. And we can actually factorize the distribution using these graphical grains. So for example, we could start from the joint distribution of the first click, the one in the red layer, and multiply this by the conditional distribution of the following clicks given the separators. Okay, so moving downwards. But of course, there is no need to start from the top. We could start from the bottom click, considering its joint, and then multiply it for the conditional distribution of the other clicks given the separators moving upwards. But of course, there is no need to start from the bottom and the top. We could start from any of the clicks of the decomposition. So to make a long story short, there is one factorization for each click which is chosen to be the root click. So in this toy example, we have 5 factorizations which are available, which are represented in this sort of matrix, where each row represents a factorization, and the darker click is the click chosen to be the root, which is then connected with the other clicks in a tree fashion, respecting the locks given by the separators. And for each such factorization, the joint distribution is simply the conditional distribution of each click given the parent clicks. Now, why do we look at all these factorizations? Because each of these factorizations give a different perspective to the joint distribution, so tells different things. And we want to exploit information to the largest extent. So for each of these factorizations, and for each click belonging to factorizations, we test the hypothesis of equality of distributions of the clicks given their parent clicks. Some of these hypotheses will be rejected, some will fail to be rejected. What we do is we collect all the clicks for which the null is rejected into a set, ending up with 5 sets. And as the very last step, we take the intersection of such sets, and this is the source set. Now, it can be proved that theoretically, probabilistically, that the estimator so defined is a consistent estimator of the source set that we defined before. And once again, we like validating our tools. So here we considered a silencing experiment because, in silencing experiments, the source of perturbation precisely, and you hope to retrieve that in your analysis.
Monica,
see?
You have 3 minutes.
Yeah, that's perfect. Thank you very much, Clara. So here we considered high-grade glioma, which is one of the most lethal brain tumors, where it is known that STAT3 is one of the master regulators. And we have a silencing experiment with 11 knockdown and 11 control samples, about 20,000 genes measured, and 17 biological pathways containing STAT3. What we hope to do is to retrieve this gene in the source set, which is actually what happens, because in 16 out of the 17 pathways, STAT3 belonged to the source set, and in 4 out of 16, it is actually the only element of the source set. So indeed, the tool seems to be working appropriately. Now, these are 2 examples of doing and saying, but of course there is much more. You can adapt graphical models to multi-omics settings, so situations in which you have measurements of different types, for example, for a gene, gene expression, copy number variation, mutation, variables of different kinds. And, graphical models are able to embrace all this heterogeneity with heterogeneous graphical models. Of course, here the level of complexity increases rapidly, both biologically and statistically speaking. But you might want to use graphical models also blindly to search for novel biologically interesting information. For example, estimating a network of interactions from scratch to search for interesting features, like, for example, hub nodes with many connections, which might have a biologically relevant impact in reality. Or you might want to use graphical models to synthesize evidence coming from different sources, maybe experiments performed in labs under partially overlapping conditions, so meta-analyzing results. Now, of course, even here, theoretically speaking, the challenges increase rapidly, but this is the reason why they are so interesting. So let me close this talk by saying that the work I've shown you is joint work with many collaborators. I hope to have listed them all in this slide. They are all smarter than me, and I wouldn't have been able to develop anything without their help. And of course, being smarter than me, they were also able to develop quite a lot of software, which is available in R, Bioconductor, and GitHub. I wouldn't have been able to do any of this. So feel free to try on the software if you are interested, and get in touch with us with positive or negative feedback if you have any. So I'm closing with some key references, the one I've mentioned along the way, and some more. And, let me thank you all for attending this talk today, but mostly for connecting with me today. So thank you very much.
Okay, thank you, Monica. Thank you, Veronica. Great talk. While we wait questions from the audience, maybe I can make a couple of questions on some issue that we have discussed many times together. The first one regards the possibility to use, pathway have connection within a pathway may have different meaning. They represent different biological connection, and sometimes may have different evidence. So the question is, do you think it is possible to use a sort of weight structure? So to use to weight the ages in the graphical framework.
Yes, this is of course a challenging question. It would be extremely appropriate in some situations to imagine a way of finding out a way of weighting the connections among nodes in a network. In the examples I've shown you, they work only in the sense of separating elements. So they work just in the sense of separating subsets of variables which are linked to conditional independencies. So it's it's a slightly different perspective. But there are some developments in the graphical modeling framework giving weights to the path between nodes. And it would be extremely interesting to exploit these in the biological setting, because the transfer of knowledge would be even larger if possible. So, but that's challenging. So that's not immediate. It requires also theoretical development. I mean, it's not just a matter of applying something applying something which is there.
Yeah. So we have some questions from the audience. Can continuous variables such as age or metabolite concentration be used instead of binary difference to elucidate members of a source set? I think this is related to. I'm not sure I've understood.
No, probably probably it was me. It wasn't particular. I wasn't particularly clear in explaining. So far, we do not have in the network any node which identifies the condition. So kind of a binary node in the network connected to all the remaining nodes to identify the condition, healthy and diseased. What we have for the time being are two different conditions separated, and we have graphical model in each condition. But of course, the source set that or the strategy that we developed to estimate the source set could be thought of in the way the person who's asking the question is figuring out. So by imagining an extra node in the network connected to the variables in the network that gives the condition, one healthy, two diseased, or the other way around. I don't know whether this answered the question, but in my understanding.
Maybe you can find, you can get in touch in the lounge.
Yeah, sure.